Good afternoon. I'm Valérie Allamand. First, I want to thank Fundación Noelia for inviting me to this event. It's a pleasure to be part of it. I have a group in the hospital Pitié-Salpêtrière, which is located in Paris. And our focus is collagen VI related dystrophies. And in particular, we're trying to develop therapeutic options for premature termination codon mutations. So first, I want to acknowledge the people who do the work on a daily basis. So this is our current group with Corinne Garciou, a research assistant, Léa Vendramini, an apprentice student, and a neurologist, the Directora Noor Bilal-Kiles, who's conducting a research project with us. And of course, past members of the group. And we are part of Gisèle Bonne's team, who some of you might know. She's an expert on laminopathies. So as you know, there are many mutations that have been reported in the genes encoding collagen VI. To date, there are over 400 mutations in COL6A1, COL6A2, and COL6A3. And at the protein level, these mutations may have different impact. And we are going to focus on this purple part here. Those are the mutations that give rise to stop codons, premature termination codons, or PTCs. And those can account for 11% of the mutations reported. So about 12 years ago, we published a study on a cohort of rare congenital muscular dystrophy patients, 49 patients. And in this cohort, we were able to establish a genotype-phenotype correlation. In that respect, patients who carry homozygous nonsense mutations, so two identical copies of a nonsense mutation, were found to be in the early onset and severe phenotype group. So we decided to focus on this group and try to give some therapeutic options to develop some therapeutic options for these patients. So when a nonsense mutation occurs in a gene, it is also it has an impact on the messenger RNA with the occurrence of a premature termination codon. And when this messenger RNA exits the nucleus to be translated into a protein, this triggers a phenomenon caused as the nonsense-mediated decay. So during the first round of translation, when the ribosome tries to read the messenger RNA into a protein, the nonsense-mediated decay recognizes that there is a premature termination codon and it degrades the transcript. So the RNA is degraded and there is no protein synthesis. And as you know, collagen VI is the product of the assembly of three chains, two short chains and one long chain that needs to assemble in a very precise way inside the cell so that they can then be secreted into the extracellular matrix. And when one of these genes carry a nonsense mutation, there is no assembly and no secretion. And this is illustrated here when we perform an immunostaining with an antibody that recognizes specifically collagen VI. So these are patient fibroblasts from a skin biopsy in culture, and we label collagen VI in red here. And what we see is that all the signal from collagen VI staining is inside the cell. And by electron microscopy, we showed that the collagen VI is retained in an intracellular compartment called the endoplasmic reticulum. On the other hand, control fibroblasts secrete normal collagen VI outside the cells in the extracellular matrix. So what can we do to correct nonsense mutations? So we have one project that is a global strategy using gene therapy to replace the defective gene. I won't present this approach today, but I will focus on the approaches that we are taking to try to inhibit the degradation of the mutant transcript by the nonsense-mediated decay, or to try to force the synthesis of the protein by inducing translational read-through. And for this, we are using pharmacological compounds, small molecules, and those projects are funded by the NOAA project and the AFM Téléthon. And we have also another approach that takes advantage of engineered tRNAs to force the synthesis of the protein. And this project is funded by the Muscular Dystrophy Association. So these three projects are collaborative projects. The NOAA project is in collaboration with Rotem Carney in Jerusalem, and he's developed or identified small molecules that are inhibitors of the nonsense-mediated decay. The AFM Téléthon project is in collaboration with two groups from France, Olivier Namy and Jean-Christophe Sintra. And they've identified, and they're also chemically improving small molecules that act as read-through inducers. And the MDA project is in collaboration with Christopher Ahern at the University of Iowa in Iowa City. And I will show you the global strategy that we take for these pharmacological approaches and some preliminary results. So our global strategy is first to test whether the compounds are toxic for the cells, so for control cells, but also for patient cells. And then when we have found an ideal dose or a dose that is not toxic, we test its efficacy. And our endpoint analysis are to quantify the transcript levels, because as you know, they are degraded by the nonsense-mediated decay. And then we look whether there is protein synthesis by different strategies, immunostaining or in-cell Western or Western blotting. And we also have to assess whether this approach can correct the dysfunctional pathways that are known in those cells, such as autophagy, apoptosis, for example. So to assess the toxicity of compounds, we take advantage of an apparatus called this IncuSite live cell analysis system. So it's basically an incubator with an integrated microscope. And when we seed cells in a well, in those plates, for example, the microscope takes images. So we have live images of the cells as they proliferate and grow. So, for example, here you can see cells 4 hours after they were seeded in the well, and the same cells 7 days after seeding. And you can see that they grew very nicely, and they have completely covered the bottom of the well. And so we call that confluence. So this is 100% confluence. And this is less than 100%. And this is our readout to assess whether the compound we're testing have an effect on the fibroblast proliferation. And here are some examples of data that we obtained with 4 different molecules that we treated the cells with different concentrations. And when cells are proliferating, when they are not treated or sham treated, you can see this graph is the graph has this curve showing that the confluence is increasing with time to reach a plateau at 100%. When the molecule is not toxic, the curve looks similar. However, when the molecule is toxic and prevents the proliferation of the fibroblast, we have these curves that are very different. Here, this is a very strong toxicity, and sometimes we have less toxicity, the cells are still able to proliferate, but at a slower rate, for example. So we do this on control fibroblasts, and we also perform the study on collagen 6 deficient fibroblasts. And in general, we've seen that these cells are more sensitive to the compounds than the control fibroblasts. And so we've had to lower the dose that we can use quite a bit. But we found some doses that are compatible with good fibroblast proliferation. And then I will just show you one example of a result that we obtained. So here we treated cells from a patient with a homozygous nonsense mutation in the COL6A2 gene. And after 7 days of treatment with 3 molecules, we were happy to see that for 2 of those molecules, we had a good stabilization of the transcripts that harbor this mutation, meaning that they were no longer degraded by the nonsense-mediated decay, or at least to a lesser extent. And this fourfold and 4.5 fold change means that we have now recovered almost 45% of the transcript, whereas cells that are not treated only have 10% of the transcript remaining. So these were nice results, and we were happy. And when we looked at those cells and we looked at the protein synthesis, with one molecule, we can detect some protein synthesis. I hope you can see the staining in red here. In blue are the cell nuclei. And when the cells are treated sham, or not treated, we don't have any collagen 6. And here we can see some collagen 6. So just so that you're not confused, the conditions are different from the images I showed you before. We don't see what's inside the cells. And so we can see that there seems to be a little secretion, and those are the control cells that are not affected by the molecule. So we are pursuing on this class of molecules, and we hope that we can achieve a higher protein synthesis that can sustain functional recovery and therapeutic benefit. And so, of course, the next step will be to translate these in vitro results in vivo. So using our mouse model, which is a mouse that we developed by introducing a nonsense mutation into the COL6A2 gene. And so at the transcript level, same as we've shown in human cells, the transcripts are very strongly degraded. We only have about 10% of remaining COL6A2 transcript in the homozygous animals. And this leads to the absence of collagen 6 secretion in skeletal muscle as well as in skin fibroblast. So these mice were characterized by a postdoctoral fellow, Alondra Solares-Perez, and this work was funded by CureCMD. And she demonstrated that as all the collagen 6 deficient mice models, these mice have a myopathic phenotype with some hallmarks of the disease, such as increased number of internalized nuclei, some increased connective tissue in skeletal muscle, so fibrosis. They also have muscle atrophy, and their muscle generate less force. And here by muscle imaging, you can see that their hind limb muscle, rear leg muscle, are smaller than their wild type or control brothers or sisters. So I will stop here for this presentation. I want to thank you for your attention. And I just want to draw your attention to the Global Registry for Collagen 6 related dystrophies. which is a registry developed at Newcastle University. I'm on the steering committee, and I know that there is a representative of Fundación Noelia on the steering committee as well, so I'm sure you're aware of this registry. Thank you again for your attention. I'll be happy to answer any questions that you might have.